Where the boy said whether they wrong the wife to the hospital. You can't go there now. And when the person Moses is expecting to go and replace the guy in Chama's house.
Okay, so can you all hear me now?
Yes, sir.
Yes, sir.
Yes,
Right. Yes, sir.
So like I was saying, apologies for the mix-up. All right. All right. So I said the mitochondrial, singular for mitochondria, would have two membranes, an outer membrane and an inner membrane. The inner membrane is thrown into folds called cristae.
So your mic is muted. We can't hear you, sir.
Can you hear me?
Yes, sir. Yes, sir.
We can hear you, sir.
Yes, sir.
How's that? All right. So between the outer membrane and the inner membrane is a space. There is the intermembrane space. Inner membrane houses proteins of the electron transport chain. Now, these proteins are made up of 4 complexes: complex 1, complex 2, complex 3, and complex 4. All right. Whenever any reducing equivalent like NADH or FAD comes into the electron transport chain, it is going to bring with it electrons and hydrogen ions. Please mute yourself. Okay, so NADH will come in at the complex 1. And when it does that, it's going to release its electron, and also the proton goes into the intermembrane space. And then the electrons will be transported across up to complex 3 and 4. Likewise, anytime the electron goes through any of the complexes, hydrogen ions are moved to the intermembrane space. For FAD, FADH2 will come in at this complex 2, and it is going to, of course, give out its electron, which will move to complex 3 and 4, and subsequently you have 2 hydrogen ions going into the intermembrane space. So for NADH2, passing through, the electron transport chain, its electron allows 3 hydrogen ions to go into the intermembrane space. But for FADH2, it is 2 hydrogen ions that goes into the intermembrane space. This is important in the formation of ATP, because as the hydrogen ions are pumped back into the matrix, the protein doing the pumping gives rise to ATP. All right. So if 3 hydrogen ions comes back into the matrix, that is, it will generate 3 ATP molecules. If 2 hydrogen ions come back into the matrix, it will generate 2 ATP molecules.
Good.
Okay.
Excuse me, sir.
Oh, I don't
Yes,
wait
please.
when I was in
Sorry?
combat.
So you said when NADH2 releases an electron, where does the proton go?
The proton goes into the intermembrane space.
They don't even know they are teaching.
Your Krebs cycle takes place in the mitochondrial matrix.
Okay.
Exactly.
Yes, sir.
All right.
Thank you, sir.
I don't understand from the mitochondria and mitochondrial part. From that part, I don't understand. You can give me a bit of explanation of that part where we are now.
Please, sir. Sorry, sir. Please, sir. Can you please go back to the complex, like talking about the complex where they go to?
Okay. Let's see. Yeah.
Thank God.
All right. So up here, you have the mitochondrion, okay, and the cristae. One of this section, just a cristae, is what we are showing here. Okay. This is the outer membrane. The space between the outer membrane and the inner membrane is called the intermembrane space. This is the inner membrane. The inner membrane houses the electron transport chain, which comprises of 4 complexes: complex 1, complex 2, complex 3, and complex 4. Glycolysis takes place in the cytoplasm, that is outside the mitochondrium. Krebs cycle takes place inside the mitochondrial matrix. So mitochondria has 2 compartments, the intermembrane space and the mitochondrial matrix. So NADH that has been reduced, okay, it becomes NADH2. It comes into the electron transport chain and gives us its electron and hydrogen. Once it enters from complex 1, okay, the electrons are given off, and those electrons will migrate through the complexes up till complex 3 and 4. And every time this electron goes through, when it passes complex 3, a proton is moved to the intermembrane space. When it passes complex 4, a proton is moved into the intermembrane space. In complex 1, a proton is moved into the intermembrane space. So if you have NADH2 coming into the electron transport chain, it can make 3 protons go into the intermembrane space. But if FADH2 comes in, since it's coming in at complex 2, that means no proton from complex 1 will go into the intermembrane space. So it's only complex 3 and complex 4, when the electron passes there, through there, that it moves the proton to the intermembrane space. So this proton in the intermembrane space is pumped back into the mitochondrial matrix. Okay? And when it does that, each time a proton comes back into the mitochondrial matrix through the F1 ATPase, it makes ATP. So if NADH2 delivers its electron and to the electron transport chain, it is capable of producing 3 protons that will come back into the matrix. So that means 3 ATP will be generated. If FADH goes and delivers the electron to the electron transport chain, 2 protons are moved to the intermediary space. And so when they come back into the mitochondrial matrix, they will generate 2 ATP molecules. So if you do your math properly, you will find that out by the time you calculate how much how many ATP, NADH2 molecules, and how many FADH molecules are generated from citric cycle, plus the GTP, it will amount to 30. Right? Also, so the matrix will have enzymes, ribozymes, DNA, RNA, mRNA, granules, fibrils, and tubules. Right? Meanwhile, the mitochondria is also involved with fatty acid oxidation, because mitochondria is the powerhouse of the cell. So the TA cycle majorly takes place in the mitochondrial matrix, all right, as well as synthesis of nucleic acids and proteins. This is a micrograph of the mitochondria, okay? And you know it has circular chromosome, all right? That's the human mitochondrial chromosome. And that chromosome resembles the chromosome because found in bacteria. And that's the basis for the endosymbiotic theory, all right? So these are what some of the, what takes place in the parts of the sites for metabolic reaction in the mitochondrion. So in the outer membrane, oxidation of epinephrine, degradation of tryptophan, which is an amino acid, fatty acids. Epinephrine is an hormone, what you call your adrenaline. Then in the inner membrane, you have the oxidative phosphorylation, which we have just looked at. In the matrix, you have the Krebs cycle, you have beta oxidation of fatty acids, you have detoxification of ammonia, and storage of calcium ions. There are other functions that mitochondria takes part of, like apoptosis, which is programmed cell death. Basically, you can have like 4 types of mitochondria. There's the aerobic mitochondrium, there's the anaerobic mitochondrium, there's the hydrogenomes and the mitosomes, all right? So they all have their differences, but they are all involved in ATP synthesis. A chief difference, critical one, will be the electron acceptor, okay? Mitochondria does semi-autonomous replication. That means the genome can replicate even if the host genome is not replicating. Okay. All right. Any questions?
Yes, sir.
Okay. Yeah.
So what are genomes?
Genome is the entire DNA content of a cell.
Okay, sir. Thank you, sir.
Yes, sir. My question, sir. Please, sir, what's the difference between the electron transport chain and the Krebs cycle, given their different locations on the mitochondrion?
Good. All right. So the Krebs cycle takes place in the matrix of the mitochondria. The reducing equivalence, that is NADH2, FADH2, that is generated, okay, has to go into the electron transport chain. So it's like you have a cup of water. The water is your cytoplasm. Okay, sir. No, is the water is the matrix, mitochondrial matrix.
Okay, sir.
Now the glass will be your cristae. That is the one that's the inner membrane that has been thrown into folds.
Okay,
So
sir.
when the reactions finish in the mitochondrial, in the matrix, the NADH2, the FADH2 generated will now be moved, okay,
Okay,
to
sir.
the glass. The glass is where the proteins that perform the job of the electron transport chain, where they are situated.
Okay, sir. Okay, sir.
All right. Okay. I thought I was sharing my slide. Okay. Let me share the second slide so that we can move on. Okay, so we need to talk about, we have talked about respiration now. Okay. I know in your primary school biology, you were taught respiration is breathing in oxygen and breathing out CO2. So, is my screen visible?
Yes, sir.
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
All right. Thank you. So we'll move on. So we'll look at photosynthesis. Photosynthesis. Okay, so I'll follow this outline. Okay. But I want to point out something. Here, oxidation is what happens in mitochondria. Photosynthesis is what happens in chloroplast. Both of them are double membrane bounded organelle. Okay, so life will not exist if there is no energy. That energy that powers life, is solar energy. All right, but the energy is not useful for cells. So it needs to be transformed into chemical energy. All right, and that's the basis for photosynthesis. So photosynthesis is when CO2, water reacts together in the presence of sunlight and chlorophyll to form sugars. That is the simplest one that you were taught. But we can break it down into two parts. There's the light reaction and the dark reaction. But please note, the dark reaction does not take place in the dark. It is just called dark because it doesn't require solar energy. All right. One other thing is, in the dark reaction is when carbon is fixed. That is, CO2 is incorporated into molecules and sugars are formed. But in the light reaction is when water is split up, okay? And then oxygen is released. So the light reaction takes place in the thylakoids. The thylakoids are like
Is what I don't know is boiling water.
so just a minute. I need to fix my power source. All right. So the light reaction takes place in the chloroplast. But basically, the molecules that do it are called chlorophyll, which you can find in the chloroplast. As well. Now the chloroplast, you find plants, although you also have photosynthetic Can you hear me?
Yes, sir.
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Sorry, sir, please, I have a question.
Ask your question.
Yeah, sorry. Talking about the dark reaction, you said necessarily does not have to be in the dark. So let's try count that with small understanding of my biology teacher in school. She said dark reaction takes place in the night. Actually, in virus.
Moment. Please repeat. Somebody
Think about the dark
Joshua,
reaction
you
now.
need to say it again, please. I can't hear you clearly.
Yeah, so talking about the dark reaction, he said it does not have to take place in the dark and give your reason for that. You noted that. Well, I was trying to remember, recollect with what my secondary school biology teacher explained. He said the dark reaction takes place in the night, making reference to stomata. You understand my question, sir? So that's what she said. Like the dark reaction takes place in the night. Alone.
This is the night. No, not night. No, okay, that's what you were told. That's what was taught, but it doesn't take place in the night. It's because it does not require solar energy. So during the light reaction, as you call it, during when water is split up, all right, there's reaction that takes place and ATP is generated. ATP is generated, NADPH is generated. These two molecules are important for the next step. All right. So the light reaction generates ATP and NADPH, which will be needed for the next phase, or what you call Calvin cycle, or what you call the dark reaction. So anytime the cell or the plant cell has ATP and NADPH, can do what you call the Calvin cycle. Yes. All right. So you have to have it in the back. Yeah, you have a. When do you have a free? You have a lab. When is your lab?
We have not been given dates.
No, on the timetable. On the timetable. Just like I don't want to rush through this.
I think April 3rd, sir.
Time on the timetable when you have lab on your timetable. There's the time a day and time allotted for lab. Can somebody check that so we can fix the next lecture for that?
Like it's Monday by 3.
Or
Monday by 3.
Or Wednesday by
That Wednesday class from biomed.
Wednesday 9.
Sorry, please, is that when practical is
Sorry? Hello?
Yes, sir.
I said Monday by 3.
Okay, sir.
We'll
But sir,
have-
we have class by 3 on Monday.
Cannot have class by 3 because on the timetable.
We have maths from 2 to 4.
Yes, that's true.
Yes.
Sir, please, Mondays are always
Maths 101 from 2 to 4.
Okay. Which day is it now? Wednesday is what? What time?
Wednesday. Maybe you should look by. Let me check.
I think Wednesday by 3.
Yes, by 3. Yes, can work.
Wednesday by 3. Is that official?
No, sir, but we don't know when.
Monday is one I know that. Official.
Yes, we have Monday by 9. I'll be 8 to 9, and Wednesday from 9 to 10, and Friday 8 to 9.
The lab time.
We don't know about the lab time, sir.
It's not on your timetable.
Not at all, sir.
No. There was a mistake on that timetable. You have a BIO 108 instead of 107. 108 is the second semester.
So maybe we'll confirm from our course advisor.
Okay, no problem. So
Okay, sir.
Wednesday 9 to 10, right?
I think so. I think so, sir. I'm not sure.
We have chemistry.
I think it's 8 to 9. Sorry, 8 to 9. We're supposed to have biology by 8 to 9. So chemistry will be 9 to 10.
Chemistry will be 9 to 10.
So biology will be 8 to 9, where we added this month.
Okay, no problem. So I'll try and yes, set up. I'll send the slides to you prior. So people can go through. And then yes.
Thank you, sir.
Thank you, sir.
Hello, sir. Hello, sir.
Yes, please. Yes.
Please, can we get the recordings?
Don't send recordings.
Slide.
Have you not been getting the recordings for the other classes? Have you been getting recordings?
Yes.
So why are you asking me a question that is obvious now? All right.
Thank you, sir.
Any other questions for me?
Yes, sir. There's a question.
Yes. Ask your question, please.
Yes, sir.
Who is asking questions?
Sir, the person I've left, sir. Don't worry, don't mind.
Okay.
I wanted to ask a question.
Yeah.
Sir, so we don't have a particular date for the lab practical now.
What do you? No. Lab date to be announced.
Okay. Thank you.
Thank you, sir.
Yeah.
Okay.
Hello, sir. Please, sir. I have a question, sir.
Yes, Joshua.
Yes, sir. So when you're talking about the crystal, like that says the electron transport chain, and through it the DNA goes, right? So from the complex one, and I was a bit confused. I was trying to get something around that movement of DNA going through the electron transport chain and moving on. So you said something.
Sorry. What is going through the electron transport chain? Hello? What did you say is going through the electron transport chain?
DNA.
Not DNA.
Protein.
No.
The
protein.
That was I did not get really. I did not really get.
Just hear me out. So you have this set of proteins. Okay? Complex 1, 2, 3, 4. Embedded in the inner mitochondrial membrane, the crystal. All right? The function of this protein is to transport electron. That electron they are transporting is coming from either NADH or FADH. Are you following?
Yes, sir.
So if NADH2 is generated, this guy is going to transfer its electron to the first complex 1. That electron is going to move from complex 1, go to cytochrome 6, then go to complex 3, complex 1, complex 2, complex 3, complex 4. Anytime an electron moves to complex 1, proton is moved to the intermembrane space. Then when it gets to complex 3, 3, another proton is moved to the intermembrane space. When it gets to complex 4, another proton is moved to the intermembrane space. FADH, on the other hand, donates its electron to complex 2. And that electron is moved from complex 2 to complex 3, where a proton is moved to the intermembrane space, and then subsequent complex 4, where another proton is moved to the intermembrane space. So if you have NADH2 being the electron carrier and the electron transport chain, it is going to move 3 protons to the intermembrane space. If it is FADH2, it's going to move 2 protons to the intermembrane space. Now, these protons, when they are in the intermembrane space, they come back into the mitochondrial matrix through a protein called FOF1 ATPase. Anytime a proton is pumped back into the mitochondrial matrix, ATP is made. So if NADH2 donates the electron, 3 protons go into the intermembrane space, 3 protons come back into the mitochondrial matrix, and so you have 3 ATP being formed. But for FADH2, 2 protons that go from complex 3 and complex 4, and 2 come back inside to the mitochondrial matrix through the FOF1 ATPase, and it's 2 ATP molecules that are formed. Is that clear?
Yes, sir. Very clear, sir.
Any other question before we go?
And sir, sorry, please. I don't know if you can introduce, when you were showing us from the diagram, is there a video you can recommend to watch it, like the movement and I don't know. The diagram,
which diagram?
Oh, you were using a diagram. There was a diagram you used to
for the mitochondria.
Yes,
for the
yes.
mitochondria.
Yes. So I was asking
Okay,
if there's a video that you can recommend for us to watch. And
video, I don't have to look for one. We'll have to look for one. Maybe I'll just out for one, and yeah, those data.
Yeah, just like the one the previous lecturer did for DNA replication. You shared a video. It was
okay. So you were watching video on his watch.
No. Like after he explained, and with the aid of the diagram, he to further gain understanding, he recommended a video that would be helpful. So I felt like it was helpful for me. So I don't know if it's possible to.
Let me go to just a minute. Hello, those of you on YouTube, do you have any questions?
The word percentage, you just see that it's just the relative error expressed as the percentage. It's just the relative error expressed
so
as
we'll meet
the
again
percentage.
next week.
So that is your relative error.